Hey everybody, welcome to the second part of Unit 9, Nervous System. Let's get started. So in this unit we're going to be talking about reflexes, and there's a couple of different types of reflexes, but they're basically operating on the same premise, and it has to do with this serial processing. So what do we mean by that? Well, it means that once a neuron has been stimulated, that information is going to go one way to another neuron, and then that may stimulate a second or a third neuron, and then maybe we end up at our target cell, or maybe we actually stimulate another neuron in the pathway. So basically with serial processing, we mean that the input's going to travel one way along a pathway, and that one neuron stimulates the next one, which stimulates the next one, until we get to the end, which is going to be our target cell. The other thing that we know about this sort of system is that it works in an all or nothing uh, uh, pattern, so that it either functions fully or it doesn't fire at all. So that's what we mean by all or nothing manner, or an all or nothing firing pattern. And the best example of serial processing is the spinal reflex. And in a real basic spinal reflex, what's happening is that you have an afferent neuron coming in, and it's going to synapse with an efferent neuron at the spinal level that is going to send a motor message back out. That is the simplest of our serial processing, and that is what we would call a spinal reflex. So yes, it all happens at the spinal level. Now eventually the nerve may get involved, the brain may get involved with this, but if we're just looking at the reflex, it all happens at the level of the spinal cord. So a reflex is a rapid automatic response to some sort of a stimulus, and these are kind of built in to help us survive, so we don't have to consciously process every emergency sort of situation. We just have automatically built in things that we're going to do when we spot danger in some way. A particular stimulus will always cause the same response if we have a reflex around that stimulus, and it's going to occur over pathways that we call reflex arcs. And these reflex arcs are going to have at least these 5 components. We're going to have a receptor of some sort that's capable of receiving the stimulus. That impulse that gets created at the receptor is going to travel along the sensory or the afferent neuron, which is going to go to the spinal cord. That is our sensory integration center, the spinal cord. And then there's going to be some sort of a motor or efferent neuron output that is going to go to a target organ that is going to actually execute the reflex for us. So here I've given you a little image that kind of puts this together. So let's say that we see a bear. So the receptor are the eyes, and that information is going to travel along a sensory neuron to the spinal cord. And here I'm showing a little interneuron. It doesn't need to be there, but there's just one there. But we could actually think of it being real basic, that this blue neuron might actually synapse with this green one, or there might be a little interneuron there. And then the motor output is going to be to cause us to run, and our skeletal muscle would be involved with that. So that's kind of an example of a reflex arc involving a real basic stimulus effect sort of a scenario. And this is clearly an example of serial processing with regard to um, I'm going to say neuronal stimulation. Now we have a couple of different types of reflexes. One of them is known as the monosynaptic reflex. Mono meaning one, and synapse means the union between neurons. So this is a scenario where we have only one synapse between the neurons. And in order to accomplish that, that means we only have two neurons that are involved in this system. Now, my image over here is going to show you an example where we have a blue sensory neuron coming in, and it's going to engage with this green interneuron, but it's also going to engage with just this red motor neuron. And that's really the example of our monosynaptic reflex, because the blue and the red would be our two neurons that are involved here. Now, this is a real basic or real rudimentary reflex, and most of these reflexes are what we call stretch reflex, and a good example of a stretch reflex is the patellar reflex. So your receptor in this case is going to be the tendon of the patella, the infrapatellar tendon, or the patellar ligament has been renamed in the last few years, but that tendon below the kneecap, you strike that and it puts a little stretch on it, and that information is going to travel along the sensory neuron, which we see here in blue, and it's going to synapse with the motor neuron, which is red, and then that output is going to go to the quad muscles, which are going to cause the quad muscles to contract and cause the leg to kick out. So that is an example of a monosynaptic reflex, just a real basic reflex. We don't have any control over it. It's hardwired into our structure, and once this receptor gets triggered, it's off and running, and that's about all there is to it. So if we have a reflex that involves only one synapse, then it's possible that we have a reflex that involves more than one synapse, and we would call that a polysynaptic reflex. And in order to accomplish the poly, we have to have at least 3 neurons in this system. So we're still going to have a sensory neuron and a motor neuron, like what we saw with the monosynaptic reflex, but we're going to have an additional, at least one additional neuron, and this will most likely be an interneuron found in the spinal cord itself. So what happens here with this sort of a scenario is that muscle groups or adjacent muscles are going to be affected. So we have a primary muscle group that we want to stimulate, but then there's going to be adjacent muscles that are also going to be affected. Most of the reflexes that are going to trigger a polysynaptic reflex, the sensory input is going to come from the skin, and it often involves combinations of flexors and extensors. So we might have inhibition of a flexor while we activate an extensor. And this tells us the story of a very important reflex that we would call the withdrawal reflex. So let's say that you were sitting around a campfire, maybe your foot got a little bit too close to the fire, and you're starting to feel the heat. So the sensory input would be coming in this way. I know they're showing it right here at the knee, but it could be coming all the way from down at the bottom of the foot. And that information is going to come up, and because it's a reflex that we're engaging, this information is going to synapse with a couple of interneurons located here in the gray matter of the spinal cord. Now, one of these is going to create an inhibition, and the other one is going to create an activation. And because what the goal is of this reflex is to get our foot out of the fire, the thing that we're going to want to do is we want to activate the hamstrings, which are our flexors, and we want to inhibit the extensors so that when we go to react, we don't activate them too and just basically make the leg go rigid. So here comes our input. It's hot, and we need to move the leg quickly, so the hamstrings will be triggered to fire, and that will cause us to bend the knee and move the foot away from the fire, and the quads will be silenced in order to ensure that the hamstrings are allowed to do their business. That is a classic example of the withdrawal reflex. And the same thing is true when we think about if you had your hand on a hot fire. In that case, we would actually be firing the flexors, and we would be inhibiting the extensors, because remember, the triceps would cause the arm to extend, and we don't want to extend the arm. We actually want to bring the arm towards the torso, towards your body, to get it away from the fire. So those are called withdrawal reflexes, and those are going to be polysynaptic. They're a little bit more complex because in this scenario we have to activate one muscle group while inactivating the antagonistic muscle group. Neurons are called nerve cells, and these are the cells capable of generating an electrical impulse. This is typically what we think of when we think of nervous tissue, although there's more to nervous tissue than just neurons. Now, neurons are kind of unique cells in the body. For one thing, they have extreme longevity. Pretty much the ones that you're born with are the ones you're going to die with, for the most part. They are amitotic, or they might be amitotic with a few exceptions. So what does that mean? Well, it means that we know that generally speaking, these cells do not divide. They don't undergo mitosis and create new ones when the old ones start to wear out, because they're kind of just going to be with us for the duration. Although we do know that in certain parts of the brain, we can grow new ones. So the way that those cells divide, they don't use mitosis, they use something known as binary fission. So they are amitotic, which is really what this word is, amitotic. But it's often pronounced amitotic. So amitotic. So generally these cells are not going to divide. All right. They have a high metabolic rate, and they require a continuous supply of oxygen and glucose. So the neurons in the brain, particularly, that's where we have a big concentration of neurons, they require glucose to be fed to it on a silver platter. And the reason why knowing that is important is because most of the bodies in the cell, most of the cells in the body have the ability to take something other than carbohydrate, something like fat, for example, or even protein, and turn it into glucose. And Mother Nature has provided the cells of the body with that capability, because if we should be in a time of starvation, the cells have an awareness of that, and cells will selectively bypass the uptake of glucose, preserving that glucose for the functioning of the brain, knowing that body cells knowing that they can tap into fat stores and create, they don't technically create glucose, but they create a molecule that gets created from glucose in order to feed themselves. I'm just going to leave it like that because that's a whole other lecture. But yeah, the brain cells require glucose and they require oxygen on a continuous basis, because they are gobbling up all of that nutritional goodness in order to do their functions. Neurons have a cell body just like any other cell does, but they're going to have one or maybe more than one processes or projections coming off the cell body. And that's kind of unique among neurons. We don't see that really with other cells. You might see other features coming off of the cell, like microvilli, villi, microvilli, or cilia, or in the case of spermaflagellum. But we don't really see the same sort of processes coming off other body cells like we do with the neuron. So we're going to look at the anatomy of a neuron here and explain what we mean by these processes. So when we talk about the cell body, another name that's used for that is soma. The word soma literally means body, and here we're using it to talk about the cell body. You could actually use the word soma to talk about your own body, because the word literally means body. Okay, but here we're going to be talking about the neuron cell body. This is the part of the cell that is making things here in the neuron. Is going to be making things here. So what kind of things does a neuron make? Well, it makes proteins. It's making repair materials for its membrane. It's making a bunch of chemicals, because remember, one of the things that neurons are doing is releasing neurotransmitters. And so the cell body will be making the precursor molecules for neurotransmitters, and those will be assembled down at the axon terminal. We'll get to that in just a little bit. We have a very large endoplasmic reticulum, located in the cell body, and the endoplasmic reticulum, rough endoplasmic reticulum in the neuron has this special substance in it that's a little bit different than what we see in the rough endoplasmic reticulum of other cells. And the way that this stains, it shows up on a slide very differently than other body cells. And so this rough endoplasmic reticulum in the neuron cell body is called the Nissl bodies. And Nissl was the French, no, not French, the German, his name was Franz Nissl. He was the German neuroanatomist who developed the stain that allowed these to become visible on a slide, allowed the rough endoplasmic reticulum of neurons to become visible on a slide. And there's a little bit about the rough endoplasmic reticulum that can tell us about the health of the neuron. So if we see a robust amount of Nissl body in a neuron, we know that the neuron is healthy and doing what it's supposed to. If we see some spotty appearance in the Nissl body, we know that the cell is damaged in some way and isn't functioning very well. So the Nissl body can be an indicator of the neuron's health. The neuron cell body is going to, of course, contain a nucleus with a nucleolus, no surprise there. There'll be some pigments that we see. In most neuron cell bodies, the plasma membrane, the outer covering, is part of the receptive region that is capable of receiving electrical impulses from other neurons. So the cell plasma membrane has the ability to conduct an impulse. Most neuron cell bodies are located in the central nervous system. Most, not all. If we have clusters of neuron cell bodies in the central nervous system, we call those clusters nuclei. If we have clusters of cell bodies located in the peripheral nervous system, we call them ganglia. And when we were looking at the anatomy of the spinal cord and talking about how the afferent neurons are coming in from the back, I pointed out that we had a dorsal ganglia there because those were the clusters of cell bodies of the unipolar neurons coming in from the periphery carrying in sensory information. We'll look at more pictures, so if you're panicking right now, don't. So these arm-like projections coming off of the cell body are what we call processes. So in the central nervous system, we're going to see both the neuron cell bodies and their processes. In the peripheral nervous system, we're going to see mostly neuron processes, and the cell bodies will be located either in the central nervous system or maybe very close to it. Now, when we bundle the processes together, we're going to call that bundle of processes either a tract or a nerve. If we're in the central nervous system, we would call it a tract, and if we're in the peripheral nervous system, we would call it a nerve. All right. Now we've got two different types of processes attached to a cell body. One of those processes is a dendrite, and the other one is an axon. And before I leave this slide, I want to put it right here that the tracts and the nerves, this is just telling us whether we're in central nervous system or peripheral nervous system. What's contained in here are specifically the axons. So let's take a look at the anatomy of a neuron. So the first thing I want to point out is that this region right through here, this is the cell body. This is where we're going to see the nucleus, and in here we're going to have the little darkly colored nucleolus, and all around it we're going to have endoplasmic reticulum, and this is what we call the Nissl body. There's going to be mitochondria here, we're going to have Golgi apparatus, and we have a bunch of vesicles. So we're going to see the same sort of organelles, many of the same organelles that we learned about in Unit 3 when we learned about cells. Now, you're going to notice at this end of the cell body, we've got a bunch of different projections, or not a bunch of different ones, but a bunch of projections coming off of this end of the cell body. these projections are called dendrites. And these dendrites, any dendrite, is a receptive region. So these little projections here, these little processes, are receiving input from maybe some sort of a sensory cell, or maybe another neuron, and that electrical signal gets conveyed to the dendrites, and that electrical impulse will wash down the surface of the dendrite over to the cell body. Okay, so these are the possible projections at this end of the cell. Now on the opposite end of this particular neuron, we've got this one single projection. It's very different than what we see with all these dendrites, and this one projection right here is called an axon. So a cell can have many dendrites, but all neurons are only going to have one axon. Now this axon has the responsibility of carrying the impulse that gets received by the dendrites. It has the responsibility of carrying that impulse away from the cell, and it's going to deliver that impulse to a target cell, which is going to be located down here at the end of the axon, at the axon terminals. So the axon terminals can be in communication with a number of different types of a number of different cells, even a number of different types of cells. That's a possibility as well. So yes, we have a single axon, which at the end can branch into 5, maybe upwards of 10 different branches. So it can have up to 10 different terminals that are making contact with 10 different cells and delivering that impulse to those 10 different cells. Now you'll notice that this axon is covered with these little blue circular things, kind of oval shaped things. These are called, well, let me back up. This is called myelin. And myelin is made by a couple of different cells. They're showing you the Schwann cell right here. The Schwann cell is a cell that makes myelin in the peripheral nervous system. And the whole purpose of covering this axon with myelin is that it allows the electrical impulse that gets created and that would travel down the axon, it allows it to travel more quickly. Because what you'll notice is that we don't have one big continuous wrap of myelin around the axon, but we have these individual little wraps leaving exposed areas in between. These are called the nodes of Ranvier. There's a gap in between the myelin, and when the axon generates an action potential, that electrical signal, it will hit this myelin and skip to the next exposed area, and then skip again, and basically end up skipping all the way down until it gets to the axon terminal. And the reason why we want the skipping is because skipping is faster than having to walk or travel all the way down every square whatever inch of this little axon. So that's what we've got here. So this is called a myelinated neuron because we see this myelin on the axon. Now there's one more important region that I want to point out, and that is this transition area right here. So we're not exactly cell body, we're not exactly axon, we're kind of in this little transition zone. The artist hasn't drawn it really well, but there's like a little bit of a small hump here, and this is known as the axon hillock. And the axon hillock is important because the axon hillock is like a gatekeeper. And if the signals that are coming in from the dendrite, all the dendrites, is strong enough, by the time it reaches the axon hillock will initiate the action potential that travels down the axon. If the impulses that are coming in from all the dendrites are not strong enough to trigger the axon hillock, then no action potential will be sent down the axon. So here's the words for all of the words I gave you when we were looking at the picture. So the dendrites, dendrites are going to be the receiving end, the receptive end of the neuron. Motor neurons, those are the ones that are carrying information away from the central nervous system out to the body. These can contain hundreds of these little projections. That's possible. Um, and they're going to basically contain the same organelles as the cell body does. So you can have some mitochondria in there, maybe some vesicles and things like that. The job of the dendrite is to convey incoming signals or messages towards the cell body, and they do this by graded potentials. Now, we haven't had that lecture yet. That is contained in this unit in the theory portion, but the graded potentials are potentials that are capable, they're basically the electrical signals that are capable of traveling short distances. So they just kind of create a little flash in the pan, and that's about it. But they're going to be important because these graded potentials have the capacity of creating an action potential, and that's really what the job of the neuron is to convey action potentials. Now, in many brain areas, we're going to see finer dendrites that are highly specialized to collect information, and they might actually contain some sort of a spine or other appendage. But basically, the thing we want you to know is that dendrites are going to be the receiving portion of our neuron. They're receiving the impulse from other neurons or some sort of a sensory cell that is aimed at triggering this neuron to send a message. Now, the axon is the other process that we talked about, and each neuron is only going to have one axon. All right, and it's going to start at the axon hillock, which is that tapered region coming off of the cell body. So we can have many, many, many dendrites, but we're always only going to have one axon. Now, in some neurons, the axons are short, or they might appear to be absent, while in other neurons, they can comprise almost the entire length of the cell. And in some parts of the body, we have axons that are over 1 meter long. That's like 3 feet. So they can be pretty long. When we have these long axons, we actually call these nerve fibers. Now, occasionally an axon will have a branch coming off of it, and those are called axon collaterals. There's still only one axon leaving the neuron cell body, but at some point the axon might have a branch that goes off. And the whole reason why we would have a collateral is because we want that neuron to communicate with many cells. Because remember, it's possible that the axon terminal will have 10 branches, and if we have a collateral with another 10 branches, then that allows one neuron to communicate with upwards of 20 different cells. Like I said, axons can branch profusely at their end. I've used the number 10, but we can have many more terminal branches than that. It says here up to 10,000. The distal nerve endings are called axon terminals. There's a lot of language that can be used here, a lot of old language. Our lectures here at Galen use specifically the words axon terminal, but you can see in older articles, you might see something like terminal boutons or axonal knobs or terminal knobs. These are all the same thing. They're those little bulbous endings at the end of an axon. Neurons can be classified based upon their structure, and their structure is going to be basically dependent upon how many processes they have. Now, we know there's always going to be only one axon, but we're going to see the greatest variation in the number of dendrites. So here we've got number 1, the multipolar neuron. This is when you have 3 or more processes. So you're going to have one axon, and you're going to have a couple of dendrites, at least 2 dendrites. Here with this multipolar neuron, we see our one axon, and then we see all of this right here as a dendrite. So this is a very characteristic shape of a multipolar neuron, and this is the neuron that we typically use when we teach you about neurons, because it has such a distinct appearance. This is the most common of our neurons and a major neuron that we're going to find in the central nervous system. Now, The other possibility is that we only have two processes. So we have one axon and one dendrite. And this is what we would call bipolar. Bi means two processes. And this is what it looks like. So one of these ends is going to be the dendrite and the other end is going to be the axon. These are rare in the human body. And there's only two places where we see something like this. One is in the retina of the eye, and the other one is in the olfactory mucosa up in the nose, which is associated with our sense of smell. So bipolars are found in the eyes and the nose, and that's it. Now the unipolar, I talked about this already, the unipolar is kind of deceiving because it does have one process coming off the cell body. But what's traveling in this one process is information coming in from the dendritic end, and then information leaving, traveling in the axonal end. So we say it's a T-like process, but there's actually two things happening here. It says here two axons. We could think of it as just really two projections wrapped up in one. Sometimes it's called a pseudo-unipolar because of the fact that you actually have two things running through that single process. Unipolar is going to be our peripheral nervous system sensory cell. So the fact that you can feel heat, thank your unipolar neuron for that. You can feel vibration. You can tell the shape of something. All of that's going to come by way of unipolar neurons. Okay. So what's going to end up happening is you're going to have a very long part that is coming off the distal end, the dendritic end, and then usually the cell bodies are located really close to the central nervous system, just outside of the spine, in one of those dorsal ganglia that we see next to the spinal cord. So there's a little bit of information that's interesting here. So we're looking at our multipolar, our bipolar, and our unipolar. And I'm going to kind of just show you the unipolar right here. So here we have the receptive end, the dendritic end. And then the thing you're going to notice is that the rest of this is classified as the axon. And it's going to be typically myelinated. But we're going to have a peripheral process and then a central process. Peripheral meaning out in the peripheral nervous system, and then this part is going to be what's entering into the central nervous system. If we compare the trigger zone for each of our three types of neurons, we see the trigger zone of the multipolar is going to be here where all the dendrites are. This green region is going to be the conducting zone where the action potential is traveling away from the cell body. And then the secretory region is going to be where the axon is going to be in communication with a target cell. If we look at our bipolar, this is the trigger zone. Look at that, it's a big trigger zone. And then we have a somewhat short conducting zone and then a comparable secretory zone where we're going to be in communication with a target cell. But look at what we have here with the unipolar. We have a very short trigger zone, a very long conducting zone, and then an expected secretory zone where we're communicating with our target cell. This is giving you just a little bit more information about the different structural classifications of the neurons. So we showed you the classic teaching multipolar neuron. This is actually more what they look like. They can be very odd. So here we've got all of these dendritic ends, little cell body, an axon. Looks like we got a little collateral action happening right there. Here we've got another interesting arrangement of dendrites synapsing with the cell body and then a single neuron. So these are multipolar, but they look different than what we've seen up to this point. These are really great examples of the bipolar. So this is the olfactory sensory cell used for sense of smell, and this is the one that we would see in the retina. So even though they're bipolar, they look a little bit different. And then here's the unipolar, pretty much as we expected. This is the receptive end. And then we have this peripheral portion that is out in the peripheral nervous system, the cell body, and then the rest of this would be in the central nervous system, or making entry into the central nervous system. Now, do I want you to be able to talk about the Purkinje cells or the pyramidal cells or anything like that? No, this is just to kind of give you an idea of how different these cells can be within these different classifications. When we were in the muscular system, we learned that a synapse occurs between a nerve and a muscle cell. And that's true, it does. But you can have a synapse that occurs between a neuron and another neuron, or maybe between a neuron and a gland. So there's a lot of ways for us to have a synapse. A synapse basically gets created by a neuron having a contact or communication with a target cell. Now, the nervous system works because we have information flowing from a neuron to another neuron, or maybe a neuron to a target cell. Neurons are functionally connected by synapses with other cells. So a synapse can be defined as a junction that mediate information transfer. Because remember, we're not having a direct contact between the neuron and its target. We're having a communication happen by way of the transference of neurotransmitters. So we have neurotransmitters coming from one neuron to another neuron, or from one neuron to some sort of an effector cell. So here we've got neuron to neuron. Here we've got neuron to the adrenal gland, would be the adrenal medulla most likely. We could have just as easily put a muscle cell here. So these are all the places where we can have a synapse. When you have neurons in series, like what we have here, you're going to have to name the neurons. So the neuron that is conducting the impulse towards the synapse is known as the presynaptic neuron. So this is the one that's sending the information. And the one that receives the information is known as the postsynaptic neuron. That's the neuron that is going to be transmitting that electrical signal away from the synapse. Now, in the peripheral nervous system, the postsynaptic structure may be another neuron, that's a possibility, or it may be some other target cell, such as a muscle cell or a gland cell. Now, the way that axons communicate with neurons communicate with each other can be rather varied. So, for example, you can have axon to dendrite communication. So this is when the axon terminals of the presynaptic neuron are connected up with the dendrites of the postsynaptic neuron. That's a possibility. You can also have axosomatic connections, which is when the axon of the presynaptic neuron is making contact with the cell body of the postsynaptic neuron. It is possible, although less common, to have axon to axon communication, or dendrite to dendrite communication, or soma to dendrite communication. But it's possible because nerves are going to do whatever they're going to do. Now, if there's one thing that we can say about nerve cells is that they are high energy, high demand cells. And as a result of that, our nerve cells have staff. They have groupies. They have people. They have cells that cater to them and ensure that their needs are met so that the neurons are functioning appropriately. So when we think about nervous tissue, then what we're thinking about are certainly the neurons, the nerve cells, and these would be basically the cells that are capable of transmitting an electrical signal. And then we're going to have all the support cells. And collectively we call these support cells neuroglia or glial cells. Okay? Now, in the central nervous system, we have 4 cells that are going to be providing support to the neurons of the brain and the spinal cord. And they're listed for you right here. But on the following slides, we're going to talk about each one. So let's go ahead and do that. Astrocytes are one of the neuroglia, and they're named astrocytes because to the early anatomists they looked very much like stars with these projections going away from their cell bodies. This is the most abundant of the neuroglia, and as the name implies, the star-shaped, we're going to see these highly branched cells. Now, where are these branches going? Well, these branches are going to cling to neurons and capillaries. And it's the capillaries that we're most interested in, because by clinging to the capillaries, they're basically going to be creating what's known as the blood-brain barrier. So astrocytes create this barrier between the blood and the central nervous system tissue. Now, this is highly prized tissue, and we don't want a pathogen that might be floating around the bloodstream to sneak out and get into our brain. And so the astrocytes are basically the bouncers at the door that are ensuring that nothing bad leaves the blood and gets into the brain. That's basically it. All right. So they play a role in exchanges between capillaries and neurons. So the exchange that they're going to be paying attention to is they're going to be looking at the ions and the various nutrients that are trying to get out of the capillary and come into brain tissue. They will allow things like glucose and oxygen to come in while being very selective about ions and anything else such as a pathogen that might also be trying to make its way out of a capillary and into brain tissue. So as a result of their interaction with the capillaries, they're going to control the chemical environment around the neuron. Okay. As a result, they can influence the functioning of the neurons. Well, we like that because they're going to basically be supportive in that way. And another thing that they are going to do, because they do have connections with not only the capillaries but also the neurons themselves, they create a support network that helps brace the tissues and guides the development of the neurons as neural pathways are being created and laid down. So what we're looking at here in this image is our astrocyte right here, and here we've got a neuron, and we can see the astrocyte has these little projections wrapping around the capillary bed, basically being very selective about what it's allowing to come out of the bloodstream and into the neural tissue. But then it's also attached to the neuron itself and is creating some structural support there. So in this way, the astrocytes provide some guidance to the way that neurons may be laying down axons, for example, creating those neural networks. So I like to think of astrocytes as the bouncers at the door of a club. Basically, they allow certain things in while keeping certain riffraff out, and make sure that the scene is kept the way that it's supposed to be. So the astrocytes are the bouncers at the door. Now, microglial cells, as the name implies, these are rather small cells, and they're actually a type of a macrophage, which is a cell of the immune system. And what these guys do is they run around and they might go to an area where you've got an injured neuron, and they'll do debris cleanup. So if you've had a neuron that's been injured or had something happen and there's a bunch of cell parts laying around, as a result of that, the microglia goes over there and makes sure that all of that gets tidied up. The other thing is that they have the ability to phagocytize any sort of microorganism that might have made its way out of the blood supply and into the brain tissue. So that's the immune function that we're looking at there. So it can also gobble up waste products of the nerves and just kind of basically just keep the environment very tidy. So where the astrocytes were the bouncers at the door, I tend to think of the microglial cells as the janitors. Now in this image they're showing the microglia or the microglial cells in contact with the axons. They don't actually wrap around like what we saw the astrocytes doing. There's no extensions wrapping around. These things will kind of migrate through the tissues a little bit, looking for areas where cleanup needs to happen. So these will move around a little bit. They're not stationary like the astrocytes are. Now, you might remember from the unit on tissues, Unit 4, we learned that epithelial cells will line all hollow cavities in the body. And believe it or not, the brain has some hollow regions in it. And the cells that line those open areas are specialized epithelial cells that we call ependymal cells. These specialized cells are going to have cilia on them, and they're also responsible for making cerebrospinal fluid. So not only do these cells help make cerebrospinal fluid, they're going to move it around with the beating of their cilia. So the things that we can say about ependymal cells is that they may be squamous, they may be columnar, they're always shown as cuboidal. For some reason, always showed as cuboidal. Most are ciliated. The cilia beats to circulate cerebrospinal fluid. These cells are responsible for maintaining the quality of the cerebrospinal fluid, and kind of in the process, they also help process the formation of cerebrospinal fluid from blood plasma. It's actually where the fluid comes from. And they form a permeable barrier between the cerebrospinal fluid and the cavities, the ventricles of the brain is what those are called, and the tissue that is being bathed by the fluid in the central nervous system. So that cerebrospinal fluid acts like a medium that will convey nutrients and signaling molecules in it. Now, the last of the cells that we're going to take a look at that are supportive in the central nervous system are the oligodendrocytes. Now, the oligodendrocytes, the name oligo means many, and dendro means branches, and cyte means cell. So this is a cell with many branches. And what this cell is doing, these branches are basically wrapping myelin around axons. So in the central nervous system, oligodendrocytes wrap axons with myelin. And it may be that one oligodendrocyte is providing myelin to many axons. So kind of similar to the astrocyte in that one cell can make a contact with many other cells in the nervous tissue, but it's making myelin and not acting as a bouncer. So what you're looking at in this image is that you've got the oligodendrocyte right here, and it's going to have these branches coming off of it, these processes. And these processes are creating the myelin that's wrapping around the axon, that's what the yellow part is, the axon of a nerve fiber. So this one particular cell looks like it is providing myelin to 1, 2, 3 neurons. Could be more. Probably is. But in this image, we're looking at 3. So what about support cells in the peripheral nervous system? Well, we have 2. One is called the satellite cell, and the other one is called the Schwann cell. Schwann cell. So satellite cells are the cells that we're going to see around the cell bodies in the peripheral nervous system. And these cells are very similar in function to astrocytes. Now, they don't create any sort of a blood-brain barrier between the cell bodies and the blood of the body, but they are protective in what they allow to get to the cell body. So they help maintain appropriate environment for the neuron cell body to function, and they're kind of helping keep some bad stuff away from it. The other cell is a Schwann cell, and this cell is very much like an oligodendrocyte in that it is producing myelin that is going around axons. Now, the difference between a Schwann cell and an oligodendrocyte is that where you might have had one oligodendrocyte creating multiple little myelin packets on an axon in the central nervous system. In the peripheral nervous system, each one of these little myelin wraps is a single Schwann cell. Just kind of a differentiation. That's not on this slide, but I think it was pointed out in the module, so I'm going to go ahead and point it out here. So here in the peripheral nervous system, each one of these is an individual Schwann cell. But if we were in the central nervous system, it's possible that maybe this cluster right here was provided by one oligodendrocyte. That's a possibility. So here's an enlarged picture of that. So here's our cell body of our unipolar neuron, and we can see that it's wrapped in these satellite cells that are protecting it, making sure everything's good for it. And then we see individual Schwann cells wrapped around, creating that myelin that's allowing this to conduct its nerve impulse very quickly. And with that, we are done.